Welcome to the deep dive. We take complex subjects, break them down, and give you the core insights you need for real understanding.
That's the goal.
And today, we are definitely doing that. We're pulling back the curtain on something absolutely fundamental to life itself, the amino acid.
The architect's blueprint, you could say.
Exactly. Just think for a second about the sheer complexity inside you right now. I mean, the elegance of it all. The way your muscles contract so effortlessly, letting you walk or, just gesture.
Or your immune system working silently, fighting off threats constantly.
Right.
It's an incredible molecular ballet.
A ballet, I like that. And at the very heart of this ballet, doing pretty much every job imaginable, are the true titans of biology.
Proteins.
Indeed. Proteins aren't just abundant. They are quite literally the most functionally diverse molecules we know of in living systems. Their versatility is just astounding.
And if you really want to understand that versatility, gain that professional level grasp on how these molecular powerhouses do so much, we can't just look at the finished building, right? No, you have to go deeper.
We have to go back to the blueprint, to the foundation. The individual bricks and beams, you might say. We need to really master their most fundamental building blocks.
And that's exactly our mission for this deep dive. We're going deep into amino acids. We'll pull out the most critical insights, figure out how these tiny units dictate the huge functions of life.
It's like, understanding every single rivet and girder before you can really appreciate the whole skyscraper.
Precisely. And by the end of this, you'll have that deep professional level grasp of biochemistry, seeing the blueprint from the ground up, moving beyond just memorizing facts to true mastery.
Absolutely. So get ready. We're going to explore their unique structure, which is actually deceptively simple in some ways.
Right. And how we classify them based on their properties.
They're fascinating electrical characteristics, too. And even how they act as nature's own built-in pH regulators, keeping our systems perfectly balanced.
It's a lot, but it's all interconnected.
We're talking about mastering the architect's blueprint for life itself. So when we call proteins the workhorses of life, that feels like it almost doesn't capture it fully. They're not just doing a job. They're kind of orchestrating the whole cellular show.
That's a great way to put it. They really are the conductors. Think about enzymes, for instance. They're the ultimate metabolic directors. They guide almost every chemical reaction in your body, breaking down food, producing energy.
Without them, metabolism just stops.
It would just grind to a halt. Life as we know it wouldn't be possible. And then you have polypeptide hormones like insulin.
Right, the messengers.
More than just messengers, really. They're sophisticated regulators. They signal huge changes across the body to keep everything in balance, that delicate homeostasis.
And movement. I mean, every single time you twitch a muscle or move an arm or leg, that contraction is all thanks to contractile proteins.
It's a beautifully choreographed molecular dance, enabling everything from a subtle blink to, a powerful jump.
It's amazing. And beyond the dynamic stuff, they also provide structure, right? Collagen comes to mind.
Absolutely essential structure. Collagen is the main protein in connective tissues. It forms the framework for bone. Think of it like the steel cables you find in reinforced concrete.
Ah, okay. Providing the structure for the other stuff.
Exactly. Collagen provides that essential support structure, and then the calcium phosphate crystals deposit precisely onto it. That's what gives bone its incredible strength and rigidity. It's truly foundational to our physical form.
And they're also our delivery service. And our defense force. Hemoglobin, the classic example.
A true icon. It's a transport protein, carrying oxygen from your lungs to every single cell. Super efficient.
And albumin, I know that's another big one in blood.
Right. Albumin is another major player. It transports all sorts of molecules, hormones, fatty acids, even drugs. Very versatile.
And for defense, we have immunoglobulins, antibodies.
Which are proteins specifically designed with incredible precision to spot and neutralize invaders, like bacteria and viruses. They're literally our body's biological SWAT team.
Okay, so if you step back, the sheer diversity is incredible. Regulation, structure, transport, defense.
It's breathtaking. And yet, despite this huge range of functions, all proteins, from the simplest enzyme to the most complex antibody, share this common structural feature. They are linear polymers. They're chains built from amino acids.
And that's where our deep dive begins, because these seemingly simple building blocks hold the secret to all that protein versatility.
Exactly. Understanding their individual properties is the absolute first step to understanding the whole picture of protein function. You can't skip this part if you want mastery.
And while today we're focused laser sharp on these basic blocks, it's worth just mentioning. The next step in this journey, which we'll tackle in future deep dives, is how these linear chains fold up.
And unique, complex, highly specific three-dimensional shapes.
Right, and it's those 3D shapes that ultimately let them do their specific jobs.
But you simply cannot appreciate that 3D masterpiece, that folded protein, without first understanding every single component, every brick, every beam, every connection. That's where we're starting here.
Okay, so proteins are the workhorses, amino acids are the foundational bricks. Let's really get into the anatomy of these blueprints. First question, how many different types are we actually dealing with in us, in humans? I've heard lots of different numbers thrown around.
Yeah, that's a good point to clarify. In nature, across all life, scientists have found over 300 different amino acids. It's quite a variety.
Wow, okay. But for us,
But for our purposes, especially when we're talking about the machinery inside mammalian cells, humans included, the focus narrows down considerably. We're primarily concerned with just 20, the standard 20 amino acids.
the standard 20. And these are the ones consistently found making up our proteins.
Yes, that they are the core players, the fundamental constituents of mammalian proteins,
Right.
the main characters in our biochemical story.
And there's a really crucial detail about those 20, isn't there? Something linking them directly to our genes.
Absolutely fundamental. These 20 standard amino acids are so significant because they are the only ones directly encoded by our DNA, by our genetic blueprint within the cell.
So the instructions for making proteins only specify these 20.
Directly, yes. That underscores their fundamental genetic importance. They are the primary, genetically specified building blocks. Now, you might hear about non-standard amino acids sometimes.
Right. What are those then?
It's crucial to know the difference. These non-standard ones are usually made by chemically modifying one of the standard 20 after it's already been put into the protein chain.
Ah, so like an edit after the fact.
Exactly. It's
Okay.
a post-translational modification. They aren't directly coded by the genes themselves, but represent a layer of fine-tuning, adding extra functionality.
Okay, so 20 core players, literally written into our genetic code. Now let's talk structure. What's the universal design? What does every single one of these 20 amino acids have in common? Their basic blueprint.
Okay, every amino acid, well, with one interesting exception we'll get to, shares this common architecture. At the very center, you have a carbon atom. We call it the alpha carbon.
The alpha carbon.
Yeah.
Okay.
And this alpha carbon is always bonded to 4 different chemical groups. Think of it like a central hub with 4 connections.
Got it. And what are those 4 crucial components? Let's break them down.
Okay, first you have a carboxyl group, written as EP.
Oh, check.
Second, a primary amino group, NH2.
Amino group NH2. Okay.
Now, here's that exception I mentioned, proline. It's the only one that doesn't have a primary amino group.
Oh, what does it have instead?
Its amino group is actually a secondary amino group. It's part of this unique, rigid ring structure that proline forms. We'll talk more about that.
Interesting. Okay, so carboxyl, amino, usually primary. What else?
A hydrogen atom, also bonded to the alpha carbon.
Simple enough. Hydrogen.
And finally, the 4th group is the distinctive side chain. This is what makes each amino acid unique. We universally call it the R group.
The R group. Right, so that's the variable part.
That's the variable part, exactly. Carboxyl group, amino group, hydrogen, and the R group, all attached to that central alpha carbon. That's the basic blueprint.
Okay, blueprint established. But what happens to these groups, the carboxyl and amino groups, at physiological pH? You know, around 7.4, the typical pH inside our bodies. Do they stay as carco and NH2?
This is where it gets really interesting and absolutely critical for understanding how proteins actually work in biology. At physiological pH, these groups ionize.
Ionize, meaning they gain or lose charge.
Exactly. The carboxyl group, H, is pretty acidic. It readily dissociates, meaning it gives up a proton, H+, and becomes a negatively charged carboxylate ion.
Okay, so the carboxyl group becomes negative. What about the amino group?
The amino group, NH2, is basic, so it does the opposite. It readily accepts a proton from the surroundings and becomes a positively charged ammonium ion, NH3 plus S.
Huh. So at the pH of our body, a free amino acid isn't actually neutral overall. It has both a positive and a negative charge at the same time.
Precisely. It exists as what we call a dipolar ion, or zwitterion. It carries both a positive charge on the amino group and a negative charge on the carboxyl group, but the overall net charge is 0.
A zwitterion. Okay. Now, what's the implication of this when these amino acids link up to form a protein? Are those charged groups still available?
That's a vital point for protein function. Once amino acids join together in that long chain, forming peptide bonds, Almost all of those alpha-carboxyl and alpha-amino NH3 plus groups get locked into forming those strong covalent peptide linkages.
So they're tied up in the backbone structure.
Mostly, yes. They're generally not available for other chemical reactions that would define the protein's unique function. They can still participate in hydrogen bonding along the backbone, which is important for structure, but they aren't the main drivers of specific function.
Okay, this brings us to a super important point then. If those backbone groups are largely involved in just linking up, what actually gives each amino acid its unique personality? What determines its specific role within that protein chain? This feels key to understanding the blueprint beyond the basics.
You've hit it exactly. It all comes down to the R factor, the R group. The specific chemical nature of that unique side chain is what fundamentally dictates the properties and the specific role an amino acid plays within a protein.
So it's the R group that determines everything.
It's the unique chemical fingerprint, the personality as you called it. And that's exactly what we use as the guiding principle for classifying them. This R group is where all the functional magic really happens.
What kind of magic are we talking about?
Well, how it interacts with water, for example. Its ability to form crucial stabilizing bonds like hydrogen bonds or even stronger ones. Even its potential to act as a sort of localized pH sensor within the protein's complex structure.
Wow.
Understanding these subtle differences in the R groups at the molecular level is what allows you to grasp how they scale up to dictate massive functional consequences in the body. That's mastery.
Okay, so it is all about the R group. That's the key to identity and function, which leads us perfectly into classification. How do we sort these 20 unique personalities, and why is this sorting so critical?
The guiding principle, as you said, is the properties of their side chains, their R groups. Primarily, we group them based on polarity. Are they nonpolar or polar?
Nonpolar versus polar, like oil versus water.
Essentially, yes. And if they are polar, we make a further distinction. Are they acidic or basic? This classification isn't just for tidiness. It's absolutely essential for predicting and, more importantly, understanding how a protein chain will fold up into its specific 3D shape.
And that 3D shape determines its function.
Exactly. So classifying by R group properties is really a predictive tool for protein behavior and function. It's fundamental.
Let's start with the first group then. Amino acids with nonpolar side chains, the oily ones you call them. What defines them, and what does that mean for how they behave in our watery bodies?
Okay, these nonpolar amino acids have R groups with a very even symmetrical distribution of electrons. Think hydrocarbons, mostly carbon and hydrogen.
So no charge separation?
Right. This means their side chains are largely unreactive chemically in terms of charge. They don't readily gain or lose protons, and crucially, they don't participate in hydrogen bonds or ionic bonds. Functionally, they behave like oil or lipids.
Oily or lipid-like. Okay.
And this property strongly promotes what we call hydrophobic interactions. Hydrophobic meaning water-faring.
Which leads to a really interesting behavior in proteins, doesn't it? Where they end up located in the protein structure seems to depend entirely on the environment, like they're trying to hide from water.
Precisely. In proteins that exist in aqueous solutions surrounded by water, which is highly polar, these nonpolar side chains have a strong tendency to cluster together. They effectively hide themselves away in the interior core of the protein.
Why do they do that?
To minimize their contact with the surrounding water molecules. It's energetically favorable. This phenomenon is called the hydrophobic effect. Think of oil droplets in water they naturally clump together to reduce their exposed surface area.
And this clustering is important for the protein's shape.
Absolutely critical. By packing into the interior of a folded protein, these nonpolar R groups are a major driving force for defining and stabilizing the protein's unique and specific three-dimensional structure. That structure, again, is fundamental to everything it does.
But what if the protein isn't just floating in water? What if it's, say, embedded in a cell membrane, which is mostly lipids, right? A hydrophobic environment. Does the rule flip?
Fantastic question. And yes, the principle holds, minimize unfavorable interactions, but the location changes dramatically. For proteins stuck within a hydrophobic environment, like a cell membrane made of lipids.
The nonpolar groups go to the outside.
Exactly. In that context, the nonpolar R groups are found predominantly on the outside surface of the protein. They interact favorably with the surrounding lipids of the membrane.
Maximizing those hydrophobic contacts?
Right. Again, minimizing unfavorable nonpolar interactions. This adaptability just highlights how crucial the R group's polarity is for dictating where a protein ends up and how it functions in different cellular compartments.
And we have a really powerful, almost tragic, real-world example of how critical a single R group's property can be, right? Sickle cell anemia. It powerfully illustrates this.
It's truly one of the most compelling clinical illustrations in all of biochemistry. Sickle cell anemia is this devastating genetic disease. It affects red blood cells, causing them to warp from their normal, flexible disc shape into rigid sickle shapes.
And that change causes all sorts of problems, blocking blood flow.
Exactly. And this dramatic, debilitating change stems from a single, tiny genetic alteration. A mutation in the gene for the beta subunit of hemoglobin A, the protein carrying oxygen in our blood.
What's the specific change?
Specifically, a polar amino acid, glutamate, which has a negatively charged R group, is replaced by a nonpolar amino acid, valine, at the 6th position in that beta subunit chain.
So just one swap, polar glutamate out, non-polar valine in.
Just one swap,
Right.
position number 6.
And that single change from a water-loving to a water-hating amino acid causes the entire hemoglobin molecule to behave differently, leading to aggregation and the deformation of the whole red blood cell. That's incredible.
It is absolutely mind-blowing. It profoundly underscores the critical impact that a single amino acid's R group, just its polarity in this case, has on the overall protein structure, its function, and ultimately human health. This isn't just some abstract concept. It has direct, life-altering clinical consequences. It's a prime example of how molecular details manifest as macroscopic disease.
It really drives home the importance of getting these details right. Now, within this nonpolar group, there's proline. We mentioned it earlier as an exception. It's often called the unique ring bearer or a structural rule breaker. What makes it so different?
Proline really does stand apart. Its defining feature is that its side chain actually loops back and forms a covalent bond with its own alpha amino nitrogen. This creates a rigid, 5-membered ring structure.
So the side chain connects back to the backbone nitrogen.
Yes. And this internal cyclization means proline has a secondary amino group, an NH group within the ring, instead of the primary NH2 group found in all other standard amino acids. Because of this, it's technically called an amino acid, not an amino acid, although it's always grouped with them.
I see. And that rigid ring structure must really affect how the protein chain can bend and fold around it, right? It sounds like it introduces a fixed kink.
It absolutely does. Proline's unique rigid geometry plays a fascinating dual role in protein structure. On one hand, it's crucial for the structure of collagen.
The structural protein we talked about.
Right. It contributes significantly to collagen's fibrous triple helical structure, giving it strength and flexibility for skin, bone, cartilage. But on the other hand, precisely because of its rigidity and the way it forces this distinct kink in the polypeptide chain,
It messes things up elsewhere.
it often acts as a structural disruptor in other types of proteins. It introduces a sharp bend that effectively interrupts the regular smooth spiral of alpha helices, which are very common structural elements in globular proteins.
So it breaks the helix.
It often does. It's like putting a sharp corner where you expected a smooth curve. This break is often functionally important, creating specific turns or loops needed for the protein's overall shape and function. It's a strategically placed rule breaker.
Okay, fascinating. Let's move on from the oily nonpolar crew to the next category. Amino acids with uncharged polar side chains. What's their defining feature and what key roles do they play?
All right, these amino acids are characterized by having R groups that carry zero net charge at physiological pH, so electrically neutral overall, in most biological settings.
But they are polar, unlike the last group.
Yes, they are polar. Their R groups contain atoms like oxygen or nitrogen that create an uneven distribution of electrons, allowing them to interact well with water. Now, it's worth noting a couple of them, cysteine and tyrosine, can actually lose a proton and become negatively charged, but only at a more alkaline pH, higher than physiological.
Okay, so generally uncharged but polar. What's their most crucial characteristic functionally?
Their defining and most vital characteristic is that their side chains contain functional groups like hydroxyls or amides that are highly capable of forming hydrogen bonds.
Hydrogen bonds. Those weaker but super important interactions for structure.
Exactly. This makes them inherently interactive with water and other polar molecules, and it makes them incredibly important for creating and stabilizing protein structures and for interactions between proteins and other molecules.
Which specific amino acids are we talking about here, and what are those hydrogen bonding groups?
We're primarily looking at serine, threonine, and tyrosine. All three have a highly polar hydroxyl group in their side chain. That adenine is an excellent hydrogen bond donor and acceptor.
Serine, threonine, tyrosine. Got it. Any others?
Yes, also asparagine and glutamine. Their side chains each contain both a carbonyl group, CO, and an amide group, NHK and H2. Both of these groups also have the necessary electronegative atoms, oxygen and nitrogen, to participate effectively in hydrogen bonds.
So lots of potential for hydrogen bonding.
Absolutely. These extensive hydrogen bonding capabilities are crucial for maintaining protein stability, for allowing proteins to bind precisely to specific targets, like substrates for enzymes, and for their overall intricate function within the cell. They form a network of stabilizing interactions.
And you mentioned cysteine earlier. It's in this uncharged polar group, but it has a really special role beyond just hydrogen bonding, right? Something involving sulfur. The disulfide bond, that sounds much stronger.
It certainly is. Cysteine is unique in this group. Its side chain contains a sulfhydryl or thiol group written as RSH. This RSH group is critically important. You often find it in the active sites of enzymes, directly participating in the catalytic reaction.
So it can be chemically reactive.
Yes, but its most famous and structurally significant role is in forming covalent crosslinks under the right conditions, specifically oxidative conditions.
Like outside the cell?
Often, yes. Two cysteine urSH groups, maybe on the same protein chain or even between two different chains, can be oxidized. They lose their hydrogen atoms and form a very strong covalent bond directly between the two sulfur atoms. This is a disulfide bond, or a cis bridge.
A sulfur bridge. And when two cysteines link up like this?
The resulting single unit, the two cysteines joined by the disulfide bond, is referred to as cysteine, C-Y-S-T-I-N-E.
Cysteine. Okay, so this isn't just a weak hydrogen bond. It's a strong covalent staple holding parts of the protein together. Can you give us an example of a protein that really relies on these?
Absolutely. Disulfide bonds are particularly vital for stabilizing the complex 3D structures of many extracellular proteins, the ones that have to function outside the controlled environment of the cell in places like the bloodstream, where conditions can be harsher.
Makes sense. They need to be tougher.
Exactly. Albumin, for instance, the most abundant protein in your blood plasma that we mentioned, transports things. Its remarkable structural integrity, its ability to maintain its precise shape and function while circulating, is heavily reliant on numerous internal disulfide bonds.
So they act like internal anchors.
Precisely. Indispensable internal anchors, preventing the protein from unfolding or falling apart. Even when subjected to the stresses of the bloodstream, they lock the folded structure in place.
Okay, structural roles, hydrogen bonding, disulfide bonds. But these uncharged polar side chains can also act as attachment points for other molecules. That sounds like a way to add even more complexity and control to proteins.
That's a very keen observation, and it's a huge point in understanding how protein function is regulated and diversified. Those polar hydroxyl groups we mentioned on serine, threonine, and, less commonly, tyrosine.
BO groups.
Yes, they can serve as highly specific attachment points for phosphate groups. This process is called phosphorylation.
Phosphorylation, I hear that term all the time in biology.
You do, because it's one of the most widespread and critical regulatory mechanisms known. Adding or removing a phosphate group often acts like a molecular on-off switch. It can dynamically activate or deactivate enzyme activity, change protein interactions, trigger signaling pathways, all in response to signals within the cell.
So it's a fundamental way cells control what proteins are doing.
Absolutely. It controls nearly every aspect of protein function in a highly dynamic way.
And what about adding sugars? Glycosylation, is that related?
Yes, exactly. Another major modification. The amide group of asparagine, that's an H2, and also the hydroxyl groups of serine or threonine can serve as attachment sites for complex oligosaccharide chains, basically sugar chains.
So proteins can get decorated with sugars?
They can. And when they do, they become glycoproteins. These sugar-coated proteins are involved in an incredible array of cellular processes. Things like cell recognition, how cells know who to stick to communication between cells, vital immune responses, even forming structural components of the extracellular matrix, the stuff that holds tissues together.
Wow, so adding sugars is another profound layer of complexity and function.
It significantly expands the protein's repertoire of roles. It's not just the amino acid sequence. These post-translational modifications, like phosphorylation and glycosylation, add entirely new dimensions to protein function.
Okay, this is fascinating. Now let's tackle the charged amino acids. We have the acidic ones and the basic ones, starting with acidic side chains, who are the key players here.
The primary acidic amino acids, the ones with side chains that readily donate protons, are aspartic acid and glutamic acid.
Aspartic acid and glutamic acid?
Okay, their distinctive feature is that their R groups each contain an extra carboxyl group and TOCO, in addition to the one on the main alpha carbon backbone.
An extra acidic group. And what happens to that extra carboxyl group at physiological pH around 7.4? Does it stay as SAO?
Nope. Just like the alpha carboxyl group, this side chain carboxyl group is quite acidic. So at physiological pH, it's fully ionized. It has readily donated its proton to the surroundings.
Meaning it becomes negatively charged.
Exactly. It exists as a negatively charged carboxylate group, ICHIA. So these amino acids, aspartic acid and glutamic acid, effectively carry a net negative charge on their side chains at physiological pH.
And because they're pretty much always ionized like that in the body.
Right. You'll often hear them referred to by their ionized names, aspartate and glutamate. They are potent proton donors in biological systems.
Okay, so acidic ones are proton donors, negatively charged at physiological pH. Logically, then, the basic amino acids must be proton acceptors and positively charged.
Spot on. The side chains of the basic amino acids readily accept cotons from the environment. The main players here are lysine and arginine.
Lysine and arginine.
At physiological pH, their R groups are fully ionized, meaning they have accepted a proton. As a result, they consistently carry a net positive charge. They're reliably positive guys in the body's normal environment.
Reliable positive charges. But then there's histidine. It's usually grouped with the basic ones, but I've heard it's kind of a special case, a pH chameleon. What makes it different?
Histidine is truly fascinating. And yes, pH chameleon is a great description. It is a weakly basic amino acid, but here's the key difference. Unlike lysine and arginine, which are almost always positively charged at physiological pH,
Histidine isn't.
free histidine, floating around on its own, is actually largely uncharged at pH 7.4. Its pKa is close enough to physiological pH that it's kind of on the fence.
Okay. But what about when it's in a protein? Does that change things?
That's the absolutely crucial part. When histidine is incorporated into a protein chain, its side chain, which contains a special ring called an imidazole ring, can readily flip between being positively charged when it picks up a proton or neutral when it loses that proton.
And what determines which state it's in?
It depends entirely on the local ionic environment right there within the protein's folded structure. Tiny shifts in the nearby pH or the charges of neighboring amino acids can influence whether histidine holds onto its proton or not.
So its charge is flexible. It adapts to its immediate surroundings within the protein. That sounds incredibly useful.
Precisely. And this unique property gives histidine a critical role in buffering within proteins. Its pKa is around 6.0.
Which is quite close to physiological pH, around 7.4.
Exactly. It's perfectly poised to either accept or donate protons right around the physiological pH range. This allows it to act like a tiny dynamic pH sensor or regulator, often right in the enzyme's active site or a key structural region.
Can you give an example?
A prime example is its vital function in hemoglobin. Histidine residues play a key role in binding and releasing protons as hemoglobin picks up and drops off oxygen, which helps buffer the pH of the blood during gas transport.
Wow. So it's buffering right where the action is.
Yes. And here's a really significant point for anyone aiming for mastery. Histidine is the only amino acid with a side chain that has a pKa near the physiological pH range.
The only one.
The only one. This unique characteristic makes it incredibly important for the catalytic mechanisms of many enzymes and for buffering systems throughout biology. Its ability to be right on that edge of protonation, deprotonation at physiological pH is key.
That's a really important takeaway. Okay, we've thoroughly dissected their structures and personalities based on the R groups. Now let's talk nomenclature. How do scientists actually refer to these efficiently? Because writing out phenylalanine or glutamine repeatedly in a long protein sequence must be incredibly tedious.
Oh, absolutely impractical. That's where the beauty of concise scientific communication comes in. Each of the 20 standard amino acids has a universally accepted 3-letter abbreviation and, even more practically, a 1-letter symbol.
3-letter and 1-letter codes.
Yes, these codes are absolutely essential for quick, efficient, and standardized communication in biochemistry and molecular biology, especially when you're writing out, comparing, or analyzing long protein sequences that can contain hundreds, even thousands of amino acids.
Makes sense. So how do we decode these symbols, especially the 1-letter ones? There must be some logic, right? Not just random letters. For our listeners aiming for mastery, knowing this shorthand is part of the language.
There is indeed a very logical hierarchical system designed for clarity and efficiency. The first rule is the simplest. If only one amino acid begins with a particular letter, then that letter is assigned as its symbol.
Easy enough. Example.
V for valine, C for cysteine, H for histidine. Simple and direct.
Okay. But obviously, multiple amino acids start with the same letter. A, G, L, T. What happens then?
Good point. In that case, priority goes to the most commonly occurring amino acid among those sharing the same initial letter. That amino acid gets the symbol.
Based on frequency. Example.
Glycine. It's much more common in proteins than, say, glutamate. Both start with G. So G is assigned to glycine.
So glutamate needs a different letter.
Exactly. This ensures the most frequent players get the most contuitive symbols. A is for alanine, most common. A, L for leucine, T for threonine, K for proline, S for serine.
Got it. Common ones get dibs on the initial. What's the next rule if the letter is taken or there's still conflict? I heard some are based on sound.
Yes. Some symbols are chosen because they phonetically evoke the sound of the amino acid name. It serves as a kind of mnemonic trick.
Like what?
F is for phenylalanine. R is for arginine. W is for tryptophan.
Tryptophan gets W. How does that work?
Yeah, that one's a bit more obscure. Some say it's from the double ring structure, a wide structure maybe, or maybe just needed a unique letter. But F for phenylalanine and R for arginine are clearer phonetic links. It's a little touch to aid recall.
Okay, so unique first letter, most common gets priority, similar sound. What's the last resort if none of those apply?
For the remaining amino acids that don't fit those criteria, a one-letter symbol is a sign that is phonetically suggestive, or, failing that, simply as close alphabetically as possible to the initial letter while still being unique.
Give me an example.
K is for lysine. It's near L in the alphabet. N is for asparagine. Contains N. Q is for glutamine. Sounds like quetiamine maybe.
Sure.
Y is for tyrosine. Contains Y. D is for aspartic acid. Aspartic. E is for glutamic acid. Gluemic. These require a bit more memorization.
Okay, so there's a system, even if some are a stretch. What about ambiguity? What if you know it's one of two related amino acids, but not exactly which one?
Good question. There are specific codes for that too. B represents S, meaning it could be either aspartic acid, D, or asparagine, N.
B for D or N.
Z represents GLX, meaning it could be either glutamic acid, E, or glutamine, Q.
Z for E or Q. Got it.
And finally, X is used when the amino acid is completely unidentified. It's a placeholder for unknown.
X for unknown.
This whole system, from three-letter codes down to one-letter symbols, including the ambiguity codes, is designed for immediate, high-level, efficient communication of complex protein sequence data. It lets professionals quickly interpret, compare, and discuss findings globally. It's the standard language.
A truly elegant and practical system. Okay, let's switch gears now to something equally fundamental, but maybe more. Visual. Mirror images. We need to talk about D and L isomers and why this matters so much in biology.
Right. This brings us to the fascinating concept of chirality. It's basically a fancy word for handedness in molecules.
Handedness. Like left and right hands.
Exactly like that. The key is the alpha carbon of the amino acid. Remember, it's bonded to 4 different chemical groups: carboxyl, amino, hydrogen, and the R group.
Yes. 4 different attachments.
That makes the alpha carbon an asymmetric, or chiral, center, because the 4 groups can be arranged in space around that carbon in 2 different ways that are mirror images of each other.
You can't superimpose them, like your hands.
Precisely. Your left and right hands look like mirror images, but you can't perfectly overlay one onto the other. They are non-superimposable mirror images. Chiral molecules are the same.
So most amino acids have this handedness because their alpha carbon has 4 different groups.
All except one. There's always an exception.
Let me guess. Glycine.
You got it. Glycine is the sole exception among the standard 20. Why? Because its R group is just another hydrogen atom.
So the alpha carbon is bonded to 2 hydrogens, a carboxyl group, and an amino group. Only 3 different types of groups.
Correct. Since it doesn't have 4 different groups attached, its alpha carbon is not chiral. It's symmetrical. Glycine doesn't have a handedness. Its mirror image is superimposable on itself.
Okay, so glycine is a chiral, but all the other 19 are chiral. They exist as these non-superimposable mirror image forms.
Exactly. And these 2 mirror image forms are called enantiomers. We specifically refer to them as the D form and the L form.
D and L enantiomers, based on...
It relates historically to their ability to rotate plane polarized light. D stood for destrorotatory, right rotating, and L for levorotatory, left rotating. Although the actual direction of rotation doesn't always match the DL designation, which is based on structural comparison to a reference molecule, glyceraldehyde. But the key point is they are distinct mirror image structures.
Like D-alanine and alanine are mirror images you can't superimpose.
Perfect example. And this difference, this chirality, has profound biological significance.
How so? Does our body use both D and L forms? It seems like that might cause confusion.
That's the critical point. Our bodies are incredibly specific. In mammalian proteins, virtually all proteins synthesized by humans and other mammals, only the L configuration of amino acids is used.
Only L amino acids. Wow.
Exclusively. Our entire protein synthesis machinery, the ribosomes, the enzymes involved, they are all exquisitely designed and have evolved to recognize and incorporate only L amino acids. It's a remarkable example of nature's specificity and chiral purity.
Why? Why only L?
Think about fitting a left hand into a left glove versus a right glove. The precise 3D shape of enzyme active sites and binding pockets requires the specific handedness of the L amino acids for proper foot and function. Using a mix of D and L would be like trying to build a complex machine with mirror image parts scattered throughout, and it just wouldn't work correctly. This uniformity is absolute key for the precise molecular recognition that defines biology.
So L amino acids are the standard for us. But I feel like I've heard about D amino acids showing up somewhere in nature, just not in our proteins. Where do we find them?
You have. And it's truly intriguing where they pop up.
Oh.
It highlights how nature uses different strategies. While not in our proteins, D amino acids are found in some specific, very important contexts.
Such as?
For instance, they are crucial components of the cell walls of many bacteria. They help provide rigidity and structure to the bacterial wall.
Bacteria use D amino acids in their walls. That's interesting. Does that have implications?
Huge implications.
Yeah.
Because we don't use D amino acids in the same way, these D amino acid containing structures in bacteria become excellent targets for antibiotics. Our own enzymes don't recognize or process them easily, but we can design drugs, like certain types of penicillin, that specifically interfere with the synthesis or structure of these bacterial walls containing D amino acids.
Clever, using their unique biochemistry against them. Anywhere else?
Yes. D amino acids are also found in some peptide antibiotics produced by microbes. Their presence can make these antibiotics more resistant to being broken down by protein-degrading enzymes, proteases, which typically only target L amino acid linkages. So using D amino acids can increase the antibiotic stability and effectiveness.
Fascinating. So they exist, they have distinct roles, just not in building our core proteins. One more thing on this. Can D and L forms interconvert? Can you flip a left hand into a right hand, biochemically speaking?
Under specific conditions, yes. There are specialized enzymes called racemases. Their specific job is to catalyze the interconversion between D and L isomers of free amino acids, not ones already in proteins. While this isn't used for our protein synthesis, bacteria use racemases, for example, to generate the D amino acids needed for their cell walls for the more common L amino acids they acquire. So interconversion is possible enzymatically.
Okay, structure, R groups, naming, chirality. This is truly a deep dive into the foundations. Now let's shift to something incredibly important for all biological systems, the pH power play. How amino acids act as acids, bases, and critically, as buffers.
Indeed. And this concept of buffering is absolutely central if you want to achieve mastery in biochemistry. It's at the heart of maintaining the stable internal environment, the homeostasis, that life depends on.
So how do amino acids fit in?
Well, remember those ionizable groups? The alpha-carboxyl group and the alpha-amino group, NH3+. Amino acids have these, both when they're free and significantly when they have ionizable side chains, like aspartate, glutamate, histidine, lysine, arginine, even when they're part of a protein chain.
So they have groups that can donate or accept protons.
Exactly. The carboxyl group is weakly acidic, proton donor, and the amino group is weakly basic, proton acceptor. This fundamental characteristic, being able to act as both an acid and a base, is called being amphoteric, and it allows them to act as physiological buffers.
Buffers, meaning they resist changes in pH.
Precisely. They help keep the pH within cells and tissues remarkably stable, which is vital because most enzymes and biological processes are extremely sensitive to pH changes. Even small shifts can drastically alter function.
Okay, quick refresher for everyone listening at a professional level. Acid is a proton donor, gives H+. Base is a proton acceptor, takes H+. But the term weak is important here, right? They aren't like strong acids, HCl, or strong bases, NaOH.
Absolutely. Critical distinction. Weak acids and bases only partially ionize or dissociate in water. They don't fully give up all their protons or accept all possible protons. They exist in an equilibrium between the protonated and deprotonated forms.
And that equilibrium is key to buffering.
It's the absolute key. Because they can exist in both forms, they can donate a proton if the environment becomes too basic, or accept a proton if it becomes too acidic, thereby minimizing the overall pH change. And we measure this acidity using the pH scale, which, as we know, is the negative logarithm of the hydrogen ion concentration. pH will look H plus bare. Lower pH means more H plus bare, more acidic. Higher pH means less H plus bare, more basic or alkaline.
Right. And to really nail down the quantitative relationship here, to predict exactly how pH changes affect these weak acids and bases, we need the Henderson-Hasselbalch equation. This feels like a cornerstone concept.
It absolutely is a cornerstone of biochemistry, physiology, pharmacology, you name it. It's your mathematical tool for understanding pH and buffering. It describes the precise quantitative relationship between the pH of a solution, the acid strength of a weak acid, represented by its pKa, and the relative amounts of the weak acid and its conjugate base.
Okay, let's unpack that. A weak acid, HA, dissociates partially into H plus and its conjugate base, A. So HA, H plus A.
Correct. And the equilibrium constant for this dissociation is Ka. Ka equals H plus AHA. It tells you how strong the acid is.
Larger Ka means stronger acid, more dissociation.
Yes. Smaller Ka means weaker acid, less dissociation. Now, biochemists often find it more convenient to work with pKa, which is simply the negative logarithm of Ka, pKa or Ka. So a smaller pKa corresponds to a stronger weak acid, and a larger pKa corresponds to a weaker weak acid.
Okay, pKa measures the inherent tendency of the group to give up its proton. How does Henderson-Hasselbalch use this?
By rearranging the Ka expression and taking the negative logarithm of both sides, you arrive at the Henderson-Hasselbalch equation. pH equals pKa plus the log of the ratio of the conjugate base concentration to the weak acid concentration.
Exactly. This equation isn't just theoretical, it's your biochemical crystal ball. It tells you that the pH of a solution containing a weak acid is determined by two things: the inherent strength of the acid, its pKa, and the ratio of its deprotonated form to its protonated form. Hey, why is having this quantitative relationship so revolutionary for understanding biology?
Because it allows us to predict and understand exactly how a buffer system will behave. It tells us, for example, at what pH the buffering will be most effective, when pH equals pKa, because log 1 equals 0. It tells us the pH range over which the buffer works well, roughly pKa and EDI1 pH unit. And it lets us calculate the exact proportions of the protonated and deprotonated forms at any given pH. This is indispensable for understanding everything from blood pH regulation to drug delivery. It's a mathematical blueprint for biochemical balance.
Okay, the equation provides the quantitative framework. Now let's talk practical application. Buffers in action. What exactly is a buffer doing, and how do amino acids fulfill this role?
A buffer, in essence, is a solution designed to resist large changes in pH when small amounts of acid or base are added. They are the shock absorbers of biological systems, maintaining that stable pH needed for life.
And they usually consist of?
Typically, a buffer system is composed of a mixture of a weak acid, HA, and its conjugate base, working together in equilibrium.
How do they actually perform this magic trick of absorbing pH shocks? What's the mechanism?
It's elegantly simple, really. If you add some strong acid, which means adding H plus ions to the buffer solution, the conjugate base component that's present in the buffer immediately reacts with those added H plus ions, snatching them up to form the weak acid, HA.
So the strong acid gets converted into a weak acid.
Effectively, yes. The damaging effect of the added H plus is minimized. Conversely, if you add a strong base, which usually means adding OH ions, the weak acid component of the buffer steps up. It donates its proton to neutralize the added base, OH plus HA, A plus H2O.
So the weak acid neutralizes the strong base.
Correct. This dynamic interplay effectively sponges up either added acid or added base, keeping the overall concentration of free H plus ions, and thus the pH remarkably stable within a narrow range.
And you mentioned there's an optimal point for this buffering where they work best.
Absolutely. A buffer exhibits its maximum buffering capacity, its strongest resistance to pH change, precisely when the pH of the solution is numerically equal to the pK of its weak acid component.
pH-pK is peak buffering.
Yes, because at that point, according to Henderson-Hasselbalch, the concentrations of the weak acid, HA, and its conjugate base are equal. You have equal amounts of the proton donor and the proton acceptor, ready to counteract additions of either base or acid.
But it still works pretty well around that point.
It does. A conjugate acid-base pair generally serves as an effective buffer when the pH of the solution is within approximately plus or minus 1 pH unit of its pK. This range, pK-1, is known as the effective buffering range. Outside this range, the buffer gets overwhelmed more easily.
Okay, so if we use the classic example of acetic acid and its conjugate base acetate, which has a pK of about 4.8.
Right. Its maximum buffering capacity is right at pH 4.8, and its effective buffering range would be roughly from pH 3.8 to 5.8.
And the Henderson-Hasselbalch equation also tells us which form the protonated acid, HA, or the deprotonated base, is more dominant at different pH values, right? That seems important too.
Crucial for understanding molecular behavior. At pH values below the pKa, the protonated acid form, HA, is the predominant species. The environment is relatively acidic, so the molecule tends to hold on to its proton.
Makes sense. And above the pK.
At pH values above the pKa, the deprotonated base form predominates. The environment is more alkaline, so the molecule is more likely to have given up its proton. Knowing which form is dominant is critical for predicting interactions, solubility, and, as we'll see, things like membrane permeability.
Perfect. Now let's apply all this directly to an amino acid. Let's use alanine as our model. It's simple, just the alpha carboxyl and alpha amino groups are ionizable. Its R group, a methyl group, is non-ionizable. How does this pH journey look as we titrate it, say, by adding base?
This is where we really see amino acids acting as natural buffers. We can analyze the titration curve of alanine just like we did for acetic acid, but alanine has two ionizable groups.
Two groups, two pK values.
Exactly. Let's start at a very low, very acidic pH, say pH 1. At this point, both the alpha carboxyl group and the alpha amino group are fully protonated. The carboxyl group is SUH neutral, and the amino group is NH3 plus positive.
So the overall net charge is +1.
Correct. We call this fully protonated form 1 of alanine. It carries a net positive charge.
Okay, 4Mi at low pH. Now what happens as we gradually raise the pH by slowly adding a strong base like NaOH? Which group gives up its proton first?
The alpha carboxyl group is the stronger acid of the two. It has the lower pK. So as we add base and the pH rises, the H group will be the first to dissociate. It gives up its proton.
And becomes the negatively charged carboxylate group.
Precisely. This transition takes us from form 1 plus 1 charge to form 2. Now, in form 2, the carboxyl group is negative, but the amino group is still protonated, NH3 plus positive.
So form 2 has both a negative charge and a positive charge.
Yes. This is that critically important dipolar ion, the zwitterion. It has both charges, but the overall net charge is 0. This form 2 is the isoelectric form of alanine.
Isoelectric meaning zero net charge. And we can use Henderson-Hasselbalch for this first dissociation step.
Absolutely. The pKa for this first dissociation, the carboxyl group, is designated pK1. The equation pH pK1 plus log, formula of form 1, perfectly describes the relationship during this phase of the titration. It lets us quantify the amounts of each form at any pH around pK1.
Okay, so we've gone from form 1 plus 1 to form 2, 0, the zwitterion, by deprotonating the carboxyl group around pK1. What's the second titratable group, and when does it give up its proton?
The second titratable group is the alpha amino group, which started as NH3 plus 3. This group is a much weaker acid than the carboxyl group.
Meaning it holds onto its proton more tightly.
Yes, it has a much smaller dissociation constant, which we designate K2, and therefore a significantly larger pK value, pK2. This means we need to reach a much higher, more alkaline pH before it willingly gives up its proton.
So as we continue adding base, eventually the NH3 plus group loses its proton.
Correct. As the pH rises well above pK1 and starts approaching pK2, the protonated amino group, NH3 plus in form 2, will release its proton, transforming into the neutral amino group, NH2. This takes us to form 3 of alanine.
And what's the charge on form 3?
In form 3, the carboxyl group is still negative, and now the amino group is NH2 neutral, so the overall net charge of form 3 is -1.
Okay, so 3 forms, form 1 plus 1, form 2, 0, form 3, n equal 1. And what are the typical pK values for these two groups in alanine? Knowing the numbers helps anchor this.
Good question. For the alpha carboxyl group, pK1, the pK is typically quite low, around 2.3 for alanine.
Very acidic.
pK1 around 2.4.
For the alpha amino group pK2, the pK is usually much higher in the alkaline range, around 9.1 for alanine.
pK2 around 9.1, big difference.
Significant difference. And these pK values precisely define the pH ranges where alanine and similar amino acids can act as buffers. So if we were to actually plot this titration on a graph pH versus amount of base added, what would we see visually? What features highlight the buffering?
The titration curve for alanine beautifully shows these two distinct dissociation steps. You'd clearly see two relatively flat regions on the curve.
Flat regions indicate buffering.
Exactly. These are the buffering regions. The first flat zone is centered around pH 2.3, pK1, where the arrow pair is resisting pH changes. The second, Higher flat zone is centered around pH 9.1, pK2, where the NH3 plus NH2 pair is providing its buffering capacity. These flat regions are where adding base causes the smallest change in pH because the buffer is actively working.
And what's chemically significant about the exact midpoint of those flat regions where pH equals pK1 or pH equals pK2?
At the exact point where pH equals pK1, or around 2.3 for alanine, you have precisely equal concentrations of form 1, the +1 form, and form 2, the 0 form Titration. They exist in dynamic equilibrium.
50-50 mix of form I and form 2 at pK1.
Right. Similarly, when the pH equals pK2, around 9.1 for alanine, you have precisely equal concentrations of form 2, the Titration, and form 3, the Meisen 1 form.
50-50 mix of form 2 and form 3 at pK2.
These points represent the centers of maximum buffering capacity for each respective group.
Okay. Now let's talk about the isoelectric point, PI. This is a really critical concept, especially for proteins and diagnostics. What exactly is PI, and how do we figure it out for alanine?
The isoelectric point, Ki, is super important. It's defined as the specific pH at which an amino acid, or more broadly, an entire protein molecule, has an overall net electrical charge of 0.
The pH where positive charges exactly balance negative charges.
Precisely. At this specific pH, the molecule won't migrate in an electric field because it has no net charge. For a simple amino acid like alanine, which only has 2 ionizable groups, the alpha-carboxyl and alpha-amino, calculating the PI is straightforward. It's simply the arithmetic average of its 2 pKa values.
Just average pK1 and pK2.
Exactly. So for alanine PI, pK1 plus pK2, that would be 2.3 plus 9.1, 2, 11.4, 2 equals 5.7.
So the PI of alanine is 5.7. And at this pH, which form of alanine is most abundant?
At the PI, pH 5.7, the zwitterionic form 2, the one with 0 net charge, is the predominant species by far. Technically, at the PI, there are also equal but very tiny amounts of form 1 plus 1 charge and form 3 charge present, perfectly balancing each other out to maintain that overall net neutrality. But the vast majority is the zwitterion.
And this PI concept isn't just theoretical math, right? It has major real-world applications, particularly in labs and clinics.
Oh, absolutely. It's a cornerstone of many biochemical techniques. Understanding the PI is critical for methods like electrophoresis, which is widely used to separate and analyze mixtures of proteins, often plasma proteins, in clinical diagnostics.
How does electrophoresis use PI?
Electrophoresis involves placing a mixture of proteins in a gel or buffer solution and applying an electric field. The proteins will then migrate through the field based on their net charge and size. Crucially, the buffer pH for the electrophoresis is usually chosen carefully relative to the PIs of the proteins you want to separate. For instance, to separate plasma proteins, the separation is often run at a pH of around 8.6. Since most major plasma proteins have PI values below 8.6, meaning they're negatively charged at pH 8.6, they will all migrate towards the positive electrode.
But they'll move at different speeds.
Exactly. They move at different rates depending on how much net negative charge they have at that pH, which depends on how far the pH is from their specific PI and also on their size and shape. This allows the different proteins to separate into distinct bands. Variations in the patterns of these bands are highly informative and can indicate various disease states, making electrophoresis a powerful diagnostic tool. Understanding PI is key to interpreting those patterns.
That's a great practical connection. So just to summarize the situation at physiological pH around 7.4, amino acids aren't just simple acids or bases, they're more versatile.
Precisely. At physiological pH, amino acids are truly amphoteric. They can act as both weak acids and weak bases. That's why we call them ampholytes. They inherently have that negatively charged carboxylate and that positively charged ammonium, NH3+, on their alpha carbon backbone. Plus, for those with ionizable side chains, like glutamate, aspartate, histidine, arginine, lysine, those side chains also contribute significantly to the overall charge and buffering capacity at physiological pH. This remarkable dual nature is exactly what allows them to play such vital, dynamic roles in buffering the delicate pH environment of life.
Okay. Now, we've seen how powerful the Henderson-Hasselbalch equation is for understanding amino acid titration and buffering. But its utility extends far beyond just amino acids, doesn't it? It's more of a universal biochemical tool.
It is incredibly versatile and absolutely fundamental. Its principles apply to any system involving weak acids and bases, which are ubiquitous in biology. For instance, consider the bicarbonate buffer system in your blood.
The main buffer keeping our blood pH stable.
Arguably the most critical physiological buffer we have, maintaining blood pH within a very narrow, life-sustaining range, around 7.35, 7.45. The Henderson-Hasselbalch equation is constantly used by physiologists and clinicians to understand how changes in the concentration of bicarbonate ion, HCO3, the conjugate base, and dissolved carbon dioxide, CO2, which forms carbonic acid, H2CO3, the weak acid, influence blood pH.
So doctors use it to understand conditions like acidosis or alkalosis.
Absolutely. It provides the quantitative framework for diagnosing and managing those potentially life-threatening conditions. Understanding that buffer system via Henderson-Hasselbalch is core clinical knowledge.
And it's even useful in pharmacology, understanding how drugs work in the body. This is where the practical application really hits home for professionals.
This is a prime example of applying fundamental biochemistry directly to clinical practice. A vast number of drugs are themselves weak acids or weak bases. The Henderson-Hasselbalch equation is absolutely essential for understanding their state of ionization in different parts of the body.
And why does their ionization state matter so much for drugs?
Because, as we touched on earlier, it dramatically affects how well they are absorbed, distributed throughout the body, metabolized, and eventually eliminated. For an acidic drug, let's call it HA, it exists in equilibrium with its charged conjugate base form, A. For a basic drug, let's call its protonated form, BH plus A, it exists in equilibrium with its uncharged base form, B.
And the charge is the key factor for getting across membranes, right? Cell walls, the gut lining.
It is absolutely crucial. Biological membranes are primarily lipid bilayers, fatty, oily barriers. Generally speaking, molecules pass through these membranes much more readily if they're in their uncharged neutral form.
Because oily membranes let oily molecules through.
Essentially, yes. Uncharged molecules tend to be more lipid-soluble, more hydrophobic, allowing them to slip through the membrane. Charged molecules, being more water-soluble, hydrophilic, are generally repelled by the lipid environment and have a much harder time crossing.
Can you give examples?
Sure. Think of aspirin. It's a weak acid, HA. It's the uncharged HA form that can easily permeate through the stomach lining or intestinal wall to be absorbed into the bloodstream. Its charged anion form, A, is pretty much stuck on the side it started on.
Okay. What about a basic drug?
Morphine is a classic example of a weak base. Its protonated form is BH plus charged. Its uncharged form is B. It's the uncharged B form that can readily pass through membranes, like the blood-brain barrier, to exert its effects. The charged BH plus form has much more difficulty crossing.
So the pH of the environment where the drug is stomach, intestine, blood directly impacts how much of the drug is in the absorbable uncharged form. This sounds critical for dosage and effectiveness.
Precisely. The effective concentration of the permeable uncharged form of any drug at its site of absorption, or at its target site, is determined by that dynamic interplay between the charged and uncharged forms. And that crucial ratio is dictated directly by two things: the pH of the local environment and the drug's own inherent pKa, its acid-base strength.
Henderson-Hasselbalch again.
Henderson-Hasselbalch is the tool that lets you calculate that ratio. So knowing the pH of the stomach, highly acidic, maybe 1.5, the intestine, more alkaline, maybe 6, 7, or blood plasma, around 7.4. And knowing the drug's pKa allows pharmacists and clinicians to predict how much of the drug will be absorbed, where it might accumulate, and how effective it's likely to be.
So understanding this equation is fundamental for optimizing drug therapy.
Absolutely essential. It helps in designing drug formulations, choosing routes of administration, predicting potential drug interactions, and understanding why certain drugs work better in certain conditions or patient populations. It's a powerful, real-world application of mastering these core biochemical principles, directly impacting patient care.
Wow. We have gone incredibly deep today. From the absolute basics of amino acid structure through their classification based on R groups, their chirality, their complex behavior in relation to pH, buffering, and even how all this impacts drug action. It's a huge amount of interconnected information.
It really is.
Yeah.
And sometimes when you absorb this much detail, it can start to feel like just a giant list of facts to memorize, which isn't the goal.
Right. The goal is mastery. True understanding. So how do we move beyond just, rote memorization, especially in a field like biochemistry where everything seems connected to everything else? How do we build that integrated understanding?
That's a critical question for anyone serious about learning this material deeply. Many people, understandably, initially see biochemistry as this vast, intimidating collection of pathways, structures, and equations. The sheer detail, while vital, can sometimes obscure the bigger picture.
You lose the forest for the trees.
Exactly. What's often missing is a kind of roadmap, a visual and conceptual tool that helps you see not just what the individual facts are, but how they all fit together logically to tell a coherent story. And this is precisely where concept maps become an incredibly powerful tool for achieving genuine mastery.
Concept maps. Okay, I've heard of these. They're a way of visually organizing relationships between ideas, almost like building a mental blueprint of the subject itself.
That's a great way to put it. It's a highly effective technique for visualizing the intricate connections and also the hierarchy between different concepts, rather than just listing them linearly like in an outline.
How do they work structurally?
In these biochemical concept maps, the material is usually presented hierarchically. The most general, overarching concepts, and the big ideas, are typically placed at the top. Then, the more specific, less general concepts are arranged logically underneath them, flowing downwards.
From general to specific.
Right. The ideal goal is for these maps to function as dynamic templates. They help you organize the information you already have, but they also provide a framework where you can easily integrate new information you learn later, fitting it into the existing structure. It's about building a flexible, adaptable, interconnected knowledge framework, not just isolated facts.
Okay, let's get specific about the anatomy of a map. What are the actual building blocks on the page, and how do they convey meaning?
All right. In these maps, concepts can be anything from abstract ideas, like, say, free energy or hydrophobic effect, to specific processes, glycolysis, phosphorylation, to actual chemical compounds, glucose, ATP, alanine. These concepts are drawn inside boxes.
Boxes for concepts. Simple enough.
Often, the size of the text or the box itself might indicate the relative importance of the concept, giving you a quick visual cue about its significance in the bigger picture. Then, lines are drawn between these boxes to show that the concepts are related in some way.
Just lines connecting related boxes.
Not just lines. This is crucial. The label written on the connecting line is what defines the specific relationship between the two concepts it links. So when you read the first concept, then the labeled line, then the second concept, it forms a valid, meaningful statement or proposition.
Ah, the line itself explains the connection, like alanine is an amino acid.
Exactly like that. Or amino acids are linked by peptide bonds to form proteins. And importantly, arrowheads are usually put on these lines to indicate the direction in which the connection should be read, guiding your understanding of the flow of information or causality.
That sounds much more informative and integrated than just a standard outline or list. And you mentioned something about crosslinks. That sounds like where it gets really powerful, breaking away from simple linearity.
Crosslinks are where concept maps truly demonstrate their power and superiority over more linear formats like flowcharts or outlines, especially for complex subjects like biochemistry. A crosslink is a connecting line with a labeled relationship, but it connects concepts located in different parts of the map.
So connecting ideas that might seem far apart in a linear presentation.
Precisely. Or, even more powerfully, crosslinks can connect a concept on the current map to a related concept that might have been presented in an entirely different map or chapter, maybe dealing with metabolism or genetics. They visually bridge different domains of knowledge.
So it's not just seeing step A leads to step B leads to step C in one pathway. It's seeing how concept X from, say, protein structure directly influences or relates to concept Y in, say, enzyme kinetics, even if they're normally taught weeks apart.
You've got it. Crosslinks explicitly highlight concepts that are central to multiple topics or disciplines within the field. This is incredibly empowering for anyone, whether you're a student prepping for a tough integrated exam like the USMLE, or a professional trying to solve a complex clinical problem that requires pulling together knowledge from different areas.
Because real-world problems aren't neatly divided into chapters.
Exactly right. High-stakes exams and real clinical situations demand the ability to synthesize information across multiple domains. Concept maps, especially with their crosslinks, train your brain to visually perceive and internalize these complex nonlinear relationships. You move far beyond just seeing a see also page XX note in a textbook. You build a truly robust, holistic, and interconnected mental model of the subject. That's a pathway to genuine mastery.
And that, listeners, is how you achieve true mastery. Not just knowing the facts, but seeing the connections. We've journeyed today from the absolute fundamental building blocks of life, amino acids.
Dissecting their unique structures, understanding how those diverse R groups dictate everything from protein folding to function, even disease.
We explored their fascinating electrical properties, their role as buffers, how the Henderson-Hasselbalch equation gives us predictive power over pH-dependent processes.
Including critical applications like understanding blood pH balance and even predicting how drugs will be absorbed and distributed in the body.
And we finished by looking at how powerful tools like concept mapping can help weave all this intricate detail into a cohesive, interconnected body of knowledge, propelling you from simply memorizing facts towards that deeper level of understanding and professional proficiency.
This deep dive, remember, wasn't just about accumulating facts. It was designed to help you understand the fundamental why and how behind the principles that govern life's processes, equipping you with that robust framework for genuine mastery, allowing you to truly think like an expert in the field.
So here's a final thought to leave you with. Building on everything we've explored, now that we have this deep understanding of the fundamental building blocks, these incredibly versatile amino acids, what surprising ways do you think protein structure and function could potentially be engineered or maybe even redesigned from scratch using this knowledge? How might we leverage these foundational insights to tackle some of the really big challenges facing us in medicine or biotechnology today? What innovations might be waiting just around the corner, built upon this mastery of the architect's blueprint? Something to ponder.